In microlab, a medium is a collection of nutrients used in the culturing of microbes for the isolation of microbial strains, maintenance of microbial collections, to aid in identification, to assist in the diagnosis of disease, or to find effective antimicrobial drugs against pathogens. Culture the culture of bacteria in culture medium. This isn't a very elegant sentence, but it does illustrate that the word culture is used as a verb to refer to the act of cultivating microbes, as a noun to refer to a medium, and as an adjective to describe the medium. Media can be liquid broth cultures in test tubes, flasks, or huge industrial tanks, or media can be solidified with thickeners such as starch, gelatin, or agar to make culture plates or culture slants. One use of media is for streaking for isolation of pure cultures on agar plates. Streaking has 4 steps. First, an inoculating loop with a sample of microorganisms is streaked back and forth across a small region of the agar surface. Then the loop is sterilized completely and used to make more back and forth streaks that cross the first set. This procedure of sterilizing the loop and dragging across the previous streaks is repeated 2 times. After some time, as we see here on this blood agar plate, dense growth occurs in the first area because there were many microbes deposited in the initial streaking. But with each streak, fewer microbes are carried by the sterilized loop across the surface of the agar. Some cells are streaked completely away from their neighbors and reproduce to form isolated colonies. Each colony derives from a colony-forming unit, which is a single cell or a small group of cells. Colonies often differ from one another. There are many media which we can categorize in a number of general ways. For instance, transport, defined, sometimes called synthetic, complex, selective, or differential. When an investigator knows precisely the kind and amount of every chemical in a medium, the medium is defined. For example, in this recipe for a medium to culture E. coli, every chemical is precisely characterized. In contrast, complex media contain imprecise amounts and kinds of chemicals. Nutrient broth and nutrient agar are complex media commonly used to culture numerous bacteria. These media contain partially digested protein and beef and yeast extracts. Again, the exact amount and type of each digestive product and the exact components and concentrations in the extracts are unknown. Defined and complex media can also be selective. Selective media are designed to enhance the growth of certain microbes, the selected ones, and inhibit the growth of others. Let's look at 2 selective media, saborodextrose and McConkey. On the left we see a saborodextrose agar plate, and on the right a nutrient agar plate. Both were inoculated with identical samples. Saboro agar selects for the growth of fungi, inhibiting the more numerous and faster-growing bacteria. The fungi are outcompeted by the bacteria on the non-selective nutrient agar. Media can be either defined or complex, and they can also be selective. Further, they can be differential. Differential media appear different when differing bacteria grow on them. Typically, the difference is seen as a color change. Examples of differential media are citrate agar and carbohydrate utilization broths. A citrate slant is a defined medium that is also selective and also differential. It selects for bacteria that can use citrate as a sole carbon source and indicates their presence by changing to a deep blue color, which is a positive result. Another differential medium is a carbohydrate utilization broth. These tubes contain a single carbohydrate, such as glucose, lactose, or sucrose, and they often have a small inverted tube, called a Durham tube, inside. Bacteria that utilize the carbohydrate produce acids that react with a color-indicating chemical. In this video, we have defined culture as the cultivation of microbes in cultures on culture media. Media include liquid broths and solid plates and slants. Streaking can isolate individual colony-forming units, which reproduce to form distinct colonies. Media are defined media when we fully know all their chemical components, or complex media when the specifics of chemistry remain unknown. Media can also be selective when they promote the growth of certain microbes and are differential when they display different results for different organisms.